Hello, everyone. My name is Bishakha, and this year I had the pleasure of mentoring Anya. As you've already heard, Anya is amazing. She's in ninth grade at Menlo. She's interested in science, and already done a lot of work around advocacy for girls, advocacy for science education. And when she started to this project, she was interested in actually doing the science, in doing theoretical work, in doing practical work in science, and delving deeper into the project. So from the first session, she was positive, motivated, really driven, and I could see that she's extremely talented. During the course of the project, she had like multiple challenges and was able to tackle those challenges head on, and with a positive attitude. So it is my pleasure to introduce Anya, and without further delay, I'll let Anya talk about her book.
Good afternoon, everyone. Thank you so much, Bishakha, for the wonderful intro. Today, I'm going to be talking to you about genetic engineering in the medical industry and more about my project. So Bishakha, my mentor, is an associate advisor at the Science Philanthropy Alliance. There, she engages with philanthropists, supporting scientific research. And there, she also offers guidance on funding decisions. She lives in San Jose and loves to spend time with her dog. Throughout this project, she's been extremely helpful to me, and her relationship has allowed me to deepen my connection with science and continue to motivate me to do more. Next slide, please. So as I mentioned, my project is on genetic engineering in the medical industry. My goal is to learn about how genetic engineering for medical use works and to conduct labs surrounding its applications. I chose this project because growing up as a young girl in Silicon Valley, I've been surrounded with tech my entire life. And as I've grown up, I've learned more and more about tech, and my love for it has grown deeper and deeper. Even though I've grown up in a tech hub, I've still found opportunities to see beauty in nature and the world around me. Through this, I've found myself thinking about the question, how and why the world works. As my understanding of this question deepens as I grow older, I've become fascinated with the intersection of technology and physiology. I picked 3 activities to focus on during my project. My first activity was surrounding research. I spent this last semester researching CRISPR, genetic engineering, DNA, RNA, and learning all the basics of it. This has helped me with my second objective, to do some laboratory experimentation. Here, I conducted a gel electrophoresis lab, which I'll go over later, with the help of Dr. Arnberg and Dr. Hasabuchi. Third, I plan on shadowing a scientist in the near future, probably within the next month, in which I'll have real-life experience. Throughout this project, my goal has been to deepen my knowledge of CRISPR tech and explore its potential applications and grapple with the ethical considerations surrounding genetic engineering. By engaging in practical experiments, conducting research, and shadowing a scientist, I aim to develop valuable skills, insights, and perspectives that will fuel my academic and personal growth. Next slide, please. Now I'm going to be showing you some of the figures I made as part of my research. So this is a cell, and in a cell, the DNA is in the nucleus, which is the little purple part you see. And within the nucleus, the DNA is organized into structures called chromosomes. Next slide, please. Chromosomes are long, thread-like structures in which DNA is wrapped around proteins. The DNA has a double helix connected by chemical bases. The DNA is made up of genes, which are specific sequences of bases that encode instructions for building proteins and controlling cellular processes. Each chromosome contains many genes, and each gene has a specific set of instructions for a particular function or trait. For example, your eye color or your susceptibility to certain diseases. DNA is like the big book of instructions, and genes are like the little chapters in between that tell each cell what to do. These processes are essential for a cell to function properly and carry out its specific roles throughout the body. Next slide, please. So where do genetic diseases come from? Genetic diseases stem from genetic mutation or genetic defects. Genetic mutation or defects is the alteration to one's DNA sequence. Mutations can be inherited or occur spontaneously. Mutations often cause abnormal function of a gene and lead to problems. So what is an example of one specific disease? Sickle cell anemia. In sickle cell anemia, red blood cells are sickle-shaped instead of round and can cause blockage in blood vessels, blocking flow of oxygen to tissues, causing pain and organ damage. Sickle cell disease is caused by a mutation in the gene that makes hemoglobin, And hemoglobin is a protein that carries oxygen in red blood cells. Next slide, please. So gene editing is the correction of these mutations by making changes to the DNA of an organism. Zeophens and talons are different gene editing processes used previously, and they use proteins to target specific DNA sequences for cutting. CRISPR, on the other hand, is also a genetic editor, but it's cheaper, more effective, and its development has revolutionized gene editing due to its simplicity, versatility, and efficiency. Next slide, please. Here is a figure. I'm going to be showing you figures I've made explaining the steps of CRISPR-Cas9 and its processes. Cas9 and the CRISPR-Cas9 consists of two parts, the Cas9 enzyme and the CRISPR technology. The Cas9 enzyme acts as molecular scissors and is capable of cutting DNA at specific locations. Within the CRISPR technology, there's a designed guide RNA that pairs with the DNA sequence that scientists want to target. These DNA sequences are often the DNAs that are mutated or have defects in them. The guide RNA directs the Cas9 protein to a precise location in the gene to make the cut. Second slide, please. Next slide, please. Thank you. The Cas9 cuts both strands at the guide RNA's location. This causes a double-stranded break in the DNA, which triggers cellular repair mechanisms. Next slide, please. Scientists then introduce modified DNA with the CRISPR-Cas9 technology and system to make precise edits. With this technology, we are now able to insert and replace specific DNA sequences, which allows for genetic modifications. Next slide, please. So how is CRISPR really applied? One example is through Casgevy, which is a newly FDA-approved drug made by CRISPR Therapeutics, which has revolutionized CRISPR cell treatment or sickle cell treatment. Casgevy corrects underlying defects and for the first time offers a potential cure. Casgevy gives hopes to millions of people, and this transformative therapy can improve the quality of life for millions. Casgevy is just one example of gene editing and its future prospects. It's an inspiration to me and the impact I want to have. Being able to help people with the intersection of technology and biology is one of my dreams, and I hope to be a part of something like this in the future one day. Next slide, please. Here, I'm going to be going through my experimental procedure in which I extracted DNA from a strawberry and was able to visualize it using gel electrophoresis. Yeah.
First, I cut the strawberries so
Oh, you can mute it because I can talk through it.
I added 1 tablespoon of my buffer solution and continued mixing the strawberries in the plastic bag.
I will
The
play it
buffer I
again.
added breaks.
That's okay, no problem. So first, I cut up the strawberries so that way I can put them into my bag and make them into a liquidy texture so it's easier for me to mix my solutions. I then add a buffer solution, which breaks the cells open to release the DNA. The cells are then separated, and the buffer also breaks down the lipids in the cell membrane and nuclei. Next, I add a peptide solution. This peptide solution is a protease-degrading cellular protein, which acts as an assistance to the buffer and allows me to once again break down the lipids in the cell membrane and nuclei. As you can see here, I'm using a coffee filter to drain out the cellular debris and isolate my sample. I then use ethanol to separate the DNA from the rest of the solution. As you can see now, I'm putting my solution into a test tube, and that white precipitate you see at the top, that's my DNA. Now, I take my DNA and split it into 2 pieces so I could use 2 alkaline buffers. I did this in hopes of achieving maximum results. Throughout this process, Dr. Armberg and Dr. Hasabuchi assisted me in conducting gel electrophoresis in the school lab. Next slide, please. Gel electrophoresis works because when you have a gel, we charge it with one side being negative and one side being positive. DNA is negatively charged, so therefore, when we put the slots containing DNA samples, as you see above, then we attach those to the negative side. And due to physics, which the class I'm in right now, The negative charges are repelled from the negative side and attracted towards the positive side. This allows for the movement of the DNA to happen and go through the gel. Then, through a different process which happens in the lab, we can visualize the DNA. Next slide, please. Unfortunately, I wasn't able to see any results despite my thorough processes. This happened because when I conducted my experiment at home, I conducted it the night before, and there was about a 10 to 12-hour gap between the time I conducted it and the time that I was actually able to be in the lab with my DNA. By this time, the DNA had probably dissolved and didn't allow us to visualize the DNA. This experience taught me, one, how difficult gene engineering is, and the challenges that many people have faced before me. But having to experience this challenge myself really brought to light the different challenges genetic engineering scientists have had to face. Next slide, please. Throughout my magical experience, I've worked on reading scientific papers and critically analyzing and interpreting them. This has been essential, and this has also been helpful through project planning. With my project planning, I learned a lot about theoretical scientific research and conceptualizing and executing projects with precision and foresight. My understanding of biotechnology flourished as I began studying the basics of DNA, RNA, cells, and genetic engineering. Throughout my mentorship, I began to unravel the complexities and applications of genetic engineering. Me and Bishakha engaged in discussions on ethical prospects of genetic engineering, and I recognized scientists' responsibility to the world as a whole. I expanded my scientific knowledge and experience by reading papers, conducting these experiments, and hopefully going to shadow a scientist in the near future. Going through this entire process has instilled a deeper appreciation for the groundbreaking work scientists accomplish, of which I hope to be a part of someday. Some of the lessons I've learned throughout this project is that communication is key. When I say communication is key, I mean there's so little time given for this project and there's so much that I've wanted to accomplish. So being able to communicate with my mentor and find different processes, I've had to utilize my time and make the most of what I can. Some of the challenges I've had is prioritizing work based on the timeline. This once again connects to my passion for science, as there was so much I wanted to cover, and there's such a limited time we had to do it. I had to prioritize what was essential to my project and make sure that we got the job done. Another challenge for me was reading scientific papers, as it was something new, and due to all the jargon in there, it was hard for me to decipher at first. Me and Bishakha worked through it, and step by step, I was able to understand the structure of a scientific paper, and now I feel fully confident that I'm able to read one. Thank you so much for listening to my presentation. I hope you learned a little bit more about what I've been working on this past few months and about genetic engineering in the medical industry. Thank you.
Thank you so much, Anya. Amazing presentation and work this semester. Do any of our audience members have any questions for Anya on her work?
I don't have a question, but I have a comment. I mean, you've done some fantastic work. I cannot imagine the amount of work you've done in such a short time, and you've explained it so well. And as you know, as you've learned, scientific experiences sometimes you don't get the results you want, and that's very normal, as Bishakha, I'm sure, told you. So congratulations to both of you.
Thank you so much. That means a lot. Yeah.
Hi, Nina. I loved your entire presentation. It's amazing. And I wondered if you knew why sickle cell is in the gene pool, why it doesn't get, kind of evolutionarized away.
Well, sickle cell is in the gene pool because it is passed down by parents, and it isn't one of the genetic mutations that just occur spontaneously. And I didn't mention it in the slides, but if both parents have a sickle cell gene, then their child is going to have, or is most likely going to have sickle cell. In rare cases, they don't, but they almost always will. And so in the gene pool, because both parents have sickle cell, it's more likely for the child to have it.
That's true. And also, if you have one gene sickle cell and one not, right? So you have the hybrid, you're more resistant to malaria. And that's why it stays in the gene pool. So it's really you know, it's a crippling disease if you get both copies, but if you get one, it gives you a biologic advantage.
Wow, that's amazing. Thank you.
Anu, can you please introduce yourself?
I am a radiologist in Arizona, and I am one of the mentors.
That's amazing. Thank you.
How much time does it need for the DNA and to test it out in the lab? Like, I know you did it the night before, but how much time did you have it show up?
To have it show up, what would have
Can you
been,
please repeat the question? Anya,
the
repeat the
question was, how much time would I need in between extracting the DNA and conducting the results in the lab? And my answer is, the most ideal situation would be for me to actually extract the DNA in lab and conduct it right away, after it's dried in the buffer. Unfortunately though, due to school and lack of direct access, I wasn't able to do that.
I have a question, and okay, maybe you can repeat this, but as Anya's current physics teacher and potential biology teacher, we just right now in biology junior year just talked about sickle cell anemia. Actually, we talked about it all year round, and that was the punchline. The punchline for the whole discovery of why that allele persists in the population is because it does give a selective advantage. So you're gonna learn so much more on top of all of this junior year, and the fact that you were able to work on this freshman year is awesome, and also should give you more like curiosity as to all these puzzle pieces, and some of them came together for you, but
Yeah.
a lot of them are probably still confusing and all over the
100%,
place,
definitely.
and they'll really come together beautifully biology junior year, so look forward to it.
I'm really excited.
Wonderful. Well, thank you so much, Anya. I know I certainly learned quite a bit from your presentation. So thank you again. And, you know, you can go have a seat and we'll invite Tessa up.
Thank you so much.
All right. Thank you.